Today I'm going to talk about immunodeficiency. And as you know that immunodeficiency can be defined as defective immunity due to inherited or acquired gene defect. Acquired gene defect by spontaneous mutation, and it is mainly X-linked. That means it affects boys more than, and it appears early during the infancy or during the first or second year of life. And this immunodeficiency can be classified into primary or secondary. Secondary could be due to external agent, for example, medicines, radiation, or chemicals. So we are going during this lecture to focus on primary immunodeficiency, and it is due to defect in leukocytes, complement components or complement regulatory proteins, and lymphocytes. A defect in leukocytes may be a genetic defect inherited, or it may be due to spontaneous mutation. For example, LAD123. LAD stands for leukocyte adhesion deficiency, and the deficiency in these molecules will lead to recurrent biogenic infection. And also Celiac or Celiac Higashi. In Celiac Higashi, actually, the cells are there. They are not absent. For example, neutrophils, T cytotoxic, and NK cells. But however, in these three cells, some granules are accumulated and they become defective. They cannot do their function and predispose the patient to Staph aureus, Pseudomonas, and other diseases. Complement component deficiency or deficiency in the complement regulatory proteins may lead to recurrent biogenic infection, may lead to systemic lupus erythematosus, or it may lead to disseminated Neisseria gonorrhea and Streptococcus pneumonia. And as can be seen here, this is red blood cell, usually surrounded by GBI anchor, and this GBI anchor allow some molecules, for example CD55, CD59, to bind, and this protect the red blood cell from destruction or being destructed by the complement. However, deficiency of this GBI may make this red blood cell possible target for destruction by the membrane attack complex. And also deficiency of some regulatory complement components, for example C1 inhibitory factor. And as I mentioned that C1 inhibitory factor, when the immune response is done, this C1 inhibitory factor will inhibit C1, so this will stop the complement. Otherwise, deficiency of C1 inhibitory factor will lead to what is so-called hereditary angioedema. And as I mentioned that the defect also, it could be in lymphocytes. And as you know that the lymphocytes, they are either B, T, or NK cells. B cells, as you know, that it produces antibodies. So any defect, or if the B cell is absent, this will lead to some immunodeficiency disorders. For example, proton hypogammaglobulinemia. Here the IgG is little. And another example is X-linked A-gamma globulinemia with absent IgG. However, in some autoimmune disorders, for example, celiac disease or gluten-sensitive enteropathy, GSE, the patient may suffer from selective IgA deficiency. And as you know that, let us now come to T lymphocyte. But before that, as you know that B cells, they mature, they originate from bone marrow and mature in the bone marrow. During this maturation, they interact with stromal cell of the bone marrow. And for B cells to detach from stromal cell, a cytokine known as interleukin number 7 will expressed. So any deficiency in this cytokine will make this B lymphocyte not able to detach from the bone marrow, so they are absent in the periphery. And now let us come to T lymphocyte. As you know that T lymphocytes, they are either or divided into T cytotoxic, T helper. T cytotoxic protect us from viral infection and from cancer. So any deficiency in T cytotoxic or any defect in T cytotoxic may predispose an individual to recurrent viral infection or to cancer. And as you know that T helper, they are subdivided into T helper 1 and T helper 2. T helper 1, as you know that they produce interferon gamma, and this interferon gamma recruit macrophages. The recruited macrophages will produce other cytokines, for example, tumor necrosis factor alpha, and this will lead to formation of a granuloma that protect an individual from getting tuberculosis. So any deficiency or defect in this cell, the individual will get tuberculosis. Other examples for the immunodeficiency are chronic granulomatosis. Chronic granulomatous, this is due to defect in phagocytic cell. And also other example is glucose 6-phosphate dehydrogenase deficiency. And now let us look at the consequences of the immunodeficiency. The consequences are opportunistic infection, tuberculosis or viral infection, and infection by extracellular bacteria. Infection by extracellular bacteria is due to deficiency or defect in B cell, in phagocytic cell, or complement components. TB or viral infection, it is due to defect in T cytotoxic and T helper 1. Thank you very much, Professor Ahmed Bolad.